Hello, everyone. Looks like we're getting people loaded in here. I'm going to ask my fellow panelists here if you could see my screen. All righty. So as everyone's coming in, I just want to do a personal thank you and welcome today to the Still's Disease Research and Clinical Trials webinar. My name is Tiffany Westrich-Robertson, and I am the CEO of International Foundation for Autoimmune Autoinflammatory Arthritis, or AiArthritis for short. And I also have joining us today, we have Dr. Rashmi Sinha from Systemic JIA Foundation. And then we will also be joined by Jeanette Bachir. I hope I said that correctly, from Sobi. And then we also have someone joining us from Avalo Therapeutics who will be logging on here shortly. So I just wanted to thank everyone for your support, and also a shout out to Karen Durant at Autoinflammatory Alliance, who has also so generously been helping to support and share this webinar today. So what are we going to be talking about? Well, this is in conjunction with Still's Disease Awareness Day, which is September 7th, and at the tail end of Autoinflammatory Arthritis, let me try that again, Autoinflammatory Awareness Month. And so we thought it was the perfect time to just combine all of these efforts together. So we're going to go ahead and get started here. And we'll start with myself giving you a little bit of an update on kind of where we are in the trials. I'm going to turn it over to Rashmi, who's going to talk a little bit more about the research. And then we're going to hear from Sobi and Avalo Therapeutics, who are going to give you a little inside information on some of the amazing research that they're doing. We'll wrap it up by talking about how to get involved in clinical trials and shared decision making. And there's always going to be room for questions here. So if you would like, once you're all in here, there is a chat option. So if you look over to the chat, you are welcome to say hello. Let us know where you're tuning in from. If you're affected by Still's disease, how is that? And you can also use the chat feature to ask questions. I am going to ask to hold off on the questions until the speakers are already started, because they may just answer that question. So we want to also make sure that the questions are related to the research, and as we're looking at the clinical trials, that we do not ask anything specific to, can I join that right now? You will have the opportunity to get connected to either of these trials when we're done. We also are recording this, and we will be sending additional materials on shared decision-making and clinical trials to those who were in attendance, as well as the link to join. So you don't need to take any notes if you don't want to, but of course you're always welcome to as well. So here we go. Let's talk a little bit about the basics of clinical trials. So really what we wanted you to just understand is that clinical trials are done because they're research studies, and they're evaluating the different interventions, the different ways that we could possibly find a new treatment, a new intervention, maybe even prevent disease activity. But all of that really has to be done in these different phases 1 through 4 of research. So what ends up happening is when an idea, a concept, a mechanism of action is identified as a possible solution to help people living with Still's disease, the researchers then need to jump in and figure out, is this intervention going to be safe? Is it effective? Is it better possibly than current standards of treatment? And then it's also based on need. So we put a little picture here of a brain and brainstorming, and we did that purposefully because your voice matters. So one of the things that happens is we see from prior treatments what's working in Still's disease, what's not working, and then we can listen from you and what's working for you and why is it not. And those type of reasons and rationale is why we need to continue the research that we're doing for Still's and other diseases, because it has the potential to change lives. So I'm not going to go into the phases 1 through 4, other than to just say they start off testing it with a few people to see if it's safe. Then the trials get bigger, and then it doesn't work. And then they get bigger, and we test more people. and find out, is that better than the existing? And then it goes into the regulatory process. So that could be the Food and Drug Administration, it could be the European Medicines Agency in Europe, and they decide if it's safe and effective to put onto market. And then sometimes there's also a phase 4 trial, and it could be testing for additional use or other ways to improve the disease. There's many other reasons why it will advance to stage 4 once it is sort of post-market, if you will. And you can also, I put a couple links on the bottom. 2 of the really great websites that teach you a little bit about clinical trials are the FDA and the EMA, and you can literally Google those with clinical trials and it'll come up with amazing information on the page. So as far, oh, sorry about that, a little cat fight in the background. So as far as why you would participate, first of all, I wanted to ask for those who are in the chat and those who are talking, how many of you have ever been in a clinical trial? And if you've been in a clinical trial, how was your experience? You can go ahead and start chatting if you'd like about clinical trials, maybe what you'd like to learn a little bit more about clinical trials, because when we follow up with you after the webinar, hopefully we'll be able to answer some more of those questions and provide you with more information. So some of the reasons why a person would participate in a trial, they could be having access issues. So perhaps they're not able to obtain the treatment that they need to manage their disease. So that is one option. Then disease control in general. There's a lot of persons, especially with Still's disease, who have not had success in their treatment and they're looking for other solutions. So it could be, I've exhausted the treatment, there's not a lot that's indicated specifically for Still's disease. And it really comes down to, why would you personally need to participate? And we hear often, I need to get my disease under control. Nothing is working. That tends to be one of the top priorities for people who are participating. But ultimately, it's your decision. And I'm going to circle back to this at the end, and we're talking about shared decision making. So we want to be educated, and that's what we're going to attempt to help you with today. Give a little bit of a basics on why we do clinical trials. We're going to turn it over to talk more about the research, see a couple of the trials, and then circle back on talking about how you make those decisions. But once we get the education, we talk about what our options are, what are your personal needs and your personal healthcare goals. And the bottom line is, don't go into this blindly. We need to be educated. We need to weigh our options. We need to talk to others about it, including our doctors, our families, anyone who is involved in our healthcare team, and then decide what is best for you. So I'm going to turn it over here to Rashmi, who is going to share her screen, and we're going to take it away. Rashmi, it's all you. And we need to unmute.
Yes, unmuted now, sharing my screen. Let me put into play mode. Okay, can you all see my screen?
Yes.
Excellent. So I'm going to talk, well, first of all, thanks to Tiffany for inviting me. And I'm extremely excited that the Stills community is getting organized. And Tiffany asked me to talk about the importance of research. So I'm going to really talk about how research and treatment really go hand in hand for rare diseases, and weaving in my own personal experience and the foundation. So first of all, who am I? I'm the mother of a child with systemic JIA, a scientist by training, and I've been an entrepreneur. And now I run a nonprofit dedicated to SJIA. And in case there is any confusion about this, I want to clarify right at the beginning that biologically speaking, systemic JIA and Stills are the same disease. It is a historical set of, choices made by scientists at different times, due to which they are known by different names, but they really are the same disease. So if I had understood this right at the beginning, I would have probably named the foundation Stills Disease Foundation, but I learned about this later, like many of you might as well. So my history is that in February 2013, my 1-year-old boy was diagnosed with SJIA. And we started learning about it a little bit. And at first we were told that, he was biologics helped the disease and he was put on some biologics. But within a year, it became clear that he was not going to go into remission with biologics, at least not immediately. And he got diagnosed with another rare complication in an already rare disease called interstitial lung disease. And it started with some clubbing in his fingers and toes. And he had repeated MAS episodes, which some of you might have experienced as well. Well, I used to read a lot of scientific papers about SJIA, and I had read this paper that patients who are diagnosed with this kind of lung disease often have a very bad outcome. And 70% of these kids were dying within 9 months from the pulmonary diagnosis. So that was the moment at which I said, well, you know, my career is not more important than this. And I started thinking about how do we actually help my child and other patients like him. So my son was patient one who we found, and then not found, who was diagnosed, and then we started the search for other patients. And now, of course, you know, I've learned a lot about rare disease organizations and how they work towards a cure. And our foundation is now part of a program with 29 other organizations. But at that point, I was just starting off without knowing any of this, with just the bet that we could help my son. So the first thing we did was that we worked with a researcher. This was at that point, I was at Stanford, going to Stanford Hospital. We worked with a researcher to organize the first research conference focused on this lung complication in SJIA. And after that, we organized several other research conferences since then, in, you know, probably about once in every 2 years about. And we what we do is that we bring together patients, researchers, clinicians, regulators from FDA and EMA, and pharma. And we have them all sit together and we try to discuss new upcoming therapies, and of course, what patient needs are, what are we seeing in the field. And we think that by having this open communication of everybody understanding each other, we can make things move faster. So that is one of our important goals. The second thing we do is that we organize these family conferences, and so far they've been really focused on systemic JIA, which is the pediatric version of the disease. Hopefully when COVID ends, we hope to go back, we'll be able to include stills as well. Our last virtual conference, we did include stills. One of the things we do during those conferences is encourage patients to participate in research, and we have collect samples, and almost every family attending the conference participates in research. And that has been a very important thing that our foundation has been able to support, where now there's biobanks with samples from these conferences, which has helped researchers understand more about the disease. Because see, in a rare disease, it can take a researcher 10 years to collect the type of samples that patients can help them collect very fast. And that helps all of us. So how do we actually help patients? And we really believe in care and research going hand in hand. So we have, I don't do all this myself. We have a network of patients who help and work in the same way with other patients. All of these patients typically have children with refractory disease, and they've learned enough about the disease to help other newly diagnosed patients. And we really believe that it's patients who can explain to each other the importance of participating in research. We've helped many patients get connected with experts, including sometimes get urgent help. And we help the community connect and build up. We used to at in-person conferences and now virtual. And one of the things we've learned by doing all this is by these samples that have been collected at all these conferences that we've organized and other ways as well, is that a lot of the refractory patients actually have high IL-18 and sometimes elevated CXCL9 as well. And these refractory patients also tend to have MAS. And there's a bunch of drugs that are currently approved for SJIA. But these patients, even after trying those biologics and trying different immunosuppressants, the IL-18, et cetera, still can remain elevated. And this chart shows, this is brought from a paper by Dr. Scott Canna, and it shows how this IL-18 is elevated in SJIA anyway, but these particular patients that I'm talking about with a history of MAS, they can have chronic elevation of IL-18, et cetera. So it seems like these are good biomarkers, and you're going to hear further about this later on in the this session. Now, coming back to the lung disease. So, we started with one patient, my child, and now we have, at least worked with 50 different families between the patients we know directly and through other doctors we work with. So we've gone, and it really, that's the really important part in rare disease, is that it's very hard to understand patterns through one patient. You really need multiple patients in order to understand patterns. And so by collecting all these patients and bringing them to doctors, we've helped everybody, and I have helped my child as well. So this is where I encourage all of you to learn about your disease, to participate in research. It's really important. And what have we learned about this disease, this lung disease, is that many of these patients have repeated MAS. They have very elevated IL-18 and CXCL9, and they have inadequate response to the IL-1 and IL-6 blocking biologics. Now, I'll be honest, I don't know much about this elevation of IL-18 and CXCL9 in the Still's disease, but there are papers showing that that's the case in Still's disease as well. And for patients with a history of MAS, it can be really helpful to try to get your IL-18 and CXCL9 measured, and you can do those as send out labs. I'll come back to that later. Now, why do research and treatment go hand in hand for a disease like SJIA? One of the reasons, and I borrowed this slide from permission by Dr. Scott Canna again, he has really a way of putting, framing these, is that SJIA is not Still's is not a unitary disease. It's really what's called a garbage can diagnosis. Diagnosis is by exclusion. It's by ruling out HLH, it's by ruling out infection. And then whatever is left, you put that in SJIA Still's. So that means that SJIA, what goes inside SJIA Still's is not one disease. It's probably a bunch of subtypes of diseases. And right now, they're all being treated in the same way. The same set of drugs are being tried on them. But we hope that we've helped identify, this first subtype, which is these highly elevated IL-18 patients. And over the next decade, maybe they'll be able to identify more and more of these subtypes. Maybe it'll depend on different organ involvement, different biomarkers, different genetics. I mean, research is advancing in many ways. So our hope is that as we collaborate with researchers, they'll be able to help identify more and more of these subtypes of SJIA and Still's. Now, coming back to, research and treatment are tightly linked in rare diseases. This is not like getting a diagnosis of, say, diabetes, which is, you can go to many different doctors, they'll have a good understanding, they'll be able to get you the right drugs and treat you. That's not the case. When you get a diagnosis like this, especially if you have refractory disease, then you really need to get involved. Here are some things I suggest to patients with SJIA, and I would recommend to all of you, find any relevant research studies, go and search for research studies, ask other patients, look out for, posts on forums about research studies. Find other similar patients, like I went hunting for patients with lung disease and trying to understand what that is about. Learn about the disease yourself. Understand your labs. And if you have refractory disease, it's really a good idea to ask your rheumatologist to order these advanced labs. Some of these are IL-18, CXCL9, S100. And that will tell you what, if your disease is still active in spite of the drugs, and help you find a biomarker to follow as you try different drugs. Find clinical trials, which this session is about. Find doctors who are interested in SJIA and Still's and help them become more of experts by referring other patients. So, and how our foundation can help. Our foundation has, go to YouTube. We have a lot of videos there for you to educate yourself about research in this disease. If you sign up for our mailing list, you can learn about the events we are organizing, etc. You're welcome to email me or others from the foundation. And many of us are on the Still's disease group, so you can find us there and ask us questions. And we're happy to help if we can help in any way. And with that, I think I've actually maybe ended early so that there's some time for questions. The acknowledgements, there's a lot of patients, so some of whom have joined us today, who work with us in doing this. And we also collaborate with a lot of researchers from not just the US, but also in Europe. And we are starting to work with them more and more. We also know some researchers in Asia, but are not actively working. In terms of our foundation, we really started off with systemic JIA, but every year we are starting to get more and more involved with stills, because if it is the same disease, and our efforts that we've done for SJIA apply to stills as well. So we hope that in this year, in the next year, we'll become more involved with this community. And I have given my contact information, and feel free to contact me. Katie, I hope I was in time, and if you have any questions, I'm happy to answer any.
Hi, Rashmi. I don't know if you can see the chat or not, but I do see somebody asking about the conferences, but I'm getting some clarity on that. Here's one. You mentioned the diagnosis by exclusion. It can be a pretty tedious process for the patient. Are we getting any closer to faster means of diagnosis?
Not yet. I think it's still a diagnosis by exclusion, though I think sometimes these biomarkers that I'm talking about, the IL-18, etc., actually can sometimes be help with diagnosis as well, because they are more elevated in SJIA stills than they are in pretty much any disease. And sometimes doctors, if they have doubts, they'll send this off. But the reality is that we still don't understand the disease clearly enough. And remember, there's subtypes in SJIA stills. You have patients with MAS, you have patients with refractory arthritis, which just does not seem to respond. There are patients with other types of lung issues. So no, we are not there yet. The reality is it's a tough process. And even then, sometimes you can start off with the SJIA stills diagnosis, and then you might learn that you actually have a NLRP4 mutation. Any other questions?
There's one mentioning something about submitting samples at the conference. I must have missed that part because I'm not 100% sure.
So I was talking about how during our family conferences, we help also have the option of patients participating in research studies. And this is something that is done with several rare disease foundations who have, who organize these conferences and also make available participating in research studies. So we've been able to do that very effectively. And something you realize is that rare disease patients are very enthusiastic about research, and they're very happy to participate. And I always say that we fight with the samples of the patients.
That's a really good point, Rashmi. For some of you who aren't familiar, that when the International Stills Disease Foundation dissolved in 2017 and asked AiArthritis to help sort of give a home to their existing resources, I started learning just how enthusiastic the stills community is, the rare disease group. They're just so passionate and enthusiastic about anything that's happening with research, and by all means, as they should be. I'm looking for more.
Yeah, there's a question about IL-18. So I don't actually know about IL-18 testing in Australia. In the US, you can do a send out order to Cincinnati Children's, and there's 1 or 2 other hospitals who are starting to test that. In Europe, I've actually been speaking with some research groups which your doctor can send out to them. One of them is in Munster, Germany, and there's another in Utrecht, Netherlands. But I don't, it's kind of a research lab that you have to send out for. And I don't know about Australia, but I'm actually glad to look into it. So if you want to ping me, let me try to find out about IL-18 testing in Australia. Another question is about the disease diverting. Yes, it is actually true. I have heard doctors say that, especially for the patients who get diagnosed very young, sometimes their systemic disease starts coming under control, and then what remains is the arthritis. On some patients, the systemic disease does not come under control and remains systemic. It's a mixed bag, and especially it's changing now that so many patients are going into remission with the typical biologics. Any other questions?
All right. Any other questions for Rashmi? We have about a minute or so left before Sobi is going to come on and do a presentation. There we go. There's one.
About genetic testing, so it depends on what tests you are doing. There are some autoinflammatory diseases which are pretty close to stills. So in that case, there's some panels that I think if you are interested, the autoinflammatory Facebook group is a very good place to learn about some genetic panels that you can do. You can also do the whole genome testing, but honestly, you need somebody who will interpret that. And I think if I was a refractory patient, my first choice would be to go in for these biomarkers and then for genetic testing. Okay. Yes. Thank you so much.
All right. Thank you so much, Rashmi. That was fantastic. And so everyone can still ask questions to Rashmi in the chat, and we are going to turn it over now to Jeanette. And Jeanette, if you'd like to share your screen.
Yes.
Righty.
Share here. And do you see my screen now?
Yes.
Okay, great. So I will be putting it in a presentation mode. So first, thanks a lot for inviting Sobi to speak into this webinar. And good morning, everyone. So today I'm presenting on behalf of a big Sobi team. I think a lot of them are already here, and I'm speaking not about still, I'm speaking about a complication in still, which is a macrophage activation syndrome. But please, before entering my speech, I would like just to say that this slide deck is for educational purpose, and the program about MAS, which is sponsored by Sobi, it's not a promotional program. So what is MAS? As we said, MAS, it stands for Macrophage Activation Syndrome. It is a disease that it is linked to overactivation of the immune system, and it is usually observed in a context of an underlying disease. So we always have an underlying disease, and in our case, it is a rheumatological disease. And this rheumatological disease could be, of course, still disease in priority, but it can also be lupus or Kawasaki or any other rheumatological disease. So what we should keep in mind that MAS is a rare disease. It's a disease that it's observed in adult, it's observed in pediatrics patient, but it is a rare disease. So for example, we observe it in around 10% of pediatric patients who present the pediatric form of still disease, SJIA, and in around 15% of the adult patients who present the still disease. So it is rare, but also what we should keep in mind that it is a life-threatening disease. And this is really very important. It is a life-threatening disease, and it is a disease with a very big, significant burden on the patient, but also on the caregiver, on the family, on everyone. So, and you will understand later on why I am saying this. So how MAS is to present clinically? We have 3 different patterns. You can see on the right part of the slide deck, the 3 different presentation of MAS. First of all, we have the classic MAS, which is the most frequent presentation. What does that mean? This means the patient, he has an underlying disease, well diagnosed, well known, well confirmed, and then later on he will present MAS as a complication of this underlying disease. This is the first form. The second form, which is observed in approximately 25%, MAS is observed at the time of the diagnosis. So the patient, when he will go to the doctor, to the clinic, he will go with a MAS symptomatology. And because MAS is always triggered by something, then the doctor will have to look about this trigger, and then he will diagnose the still disease at a later time point. And then we have this persisting relapsing MAS. This is a form of MAS that Rashmi spoke about in her speech, and it is very rare, and it is a MAS that it continues appearing and appearing and appearing in a patient life who has the underlying disease well diagnosed and confirmed. So when MAS it occurs, we know that it will attack a lot of our organ. It can attack the lung, it can attack the liver, it can attack the neurological system. So clinically, usually it is very difficult to diagnose MAS, and it can be easily confused with the underlying disease. However, there are still some typical symptoms that you should be aware about that can help you to make a kind of differentiation. First of all, for example, the fever that we observe in MAS, it's not really the fever that we observe in the underlying still disease, because in MAS the fever is very high. It's a persistent fever. It's a non-remitting fever. In MAS, we have rash, and the rash is usually different from what we observe in the underlying disease because it's related to the low platelet and to the resulting bleeding. So it's the patient will present, for example, with some mucosal bleeding, with some skin bleeding, with some very easy bruising. And of course, depending on what organ will be impacted, then the patient will present the symptomatologies that can correspond. And what I want you really to keep in mind is that when MAS is here, MAS is a quickly deteriorating disease. It means within hours or even within a day, the patient or the outcome of the patient can be really very severe. And I hate to say it, but MAS can take a very fatal course very quickly and very rapidly. So it is very essential, and it is very important that when we think about MAS, that we call our doctor and we work on a diagnosis immediately with him, and that we start a treatment. This is an emergency. We need to start that treatment very, very quickly. So how the MAS is diagnosed, and this is a good thing, this is a good point, because we have approved criteria for the diagnosis of MAS. These criteria have been reviewed by rheumatological expert, and they have been approved within the ULAR ACR consortium. And these criteria are actually a combination of clinical parameters and laboratory parameters. The clinical parameters are the presence of the fever, and the laboratory parameters are based on a lab test, which is very simple to be done. It can be done in practically in every laboratory. It's based on a combination of some abnormal lab tests, as they are stated here on these slides. And usually it's a very simple test. It requires a small amount of blood. And then your doctor, according to how much of these lab tests are abnormal and how much they are abnormal in combination with the presence of fever, he will be able to tell you, yes, it's MAS, or maybe it's MAS, or no, it's not MAS. So MAS is diagnosed, MAS is here. As we said, it's imperative that we start the treatment. And what are the treatment? What are our options? Unfortunately, today, we do not have any specific treatment approved. Actually, all what we know about the treatment of MAS is really based on expert opinion, on what has been published in literature from case reports or retrospective surveys or retrospective studies. What we know is that there are drugs that work. We should not be saying no. Some of these drugs are glucocorticosteroids that are needed to be started with a very high dose, and that they are adopted actually worldwide as a mainstay treatment of MAS. Now, when the patient is not responding to glucocorticosteroid, of course, there are other treatments that are used. These treatments are called biologics. They will, some of them may work on the inhibition of interleukin-1 or interleukin-6. We know that this inhibition works very well in the underlying disease and very effective in the underlying disease. We have observed also some role of these drugs in the treatment of MAS, but not in all, maybe not in all patients, or maybe not at one single dose. So despite this progress in treatment and despite all what we know now about the treatment of MAS continue actually to present a high unmet medical need, and we still observe actually a high mortality and a high morbidity. So for example, when patient is diagnosed with MAS, and he can reach a mortality rate of 30%. And I think it's a mortality rate in a, of 30%. I think you all agree with me, it's still very high. These patients also, when they are diagnosed, and because of how much the disease is quickly deteriorating, then these patients still require in a high rate a ICU admission. They still require in a high rate oxygenotherapy or mechanical ventilation. They still present cardiovascular dysfunction and require cardiovascular support. So all this, you can understand how much it's a burden on the patient to be in ICU, for example, on how much it's a burden on a family and on a caregiver as well. So in the light of all this, we all think we need something new, but what can be this new? What can be this new drug? So actually what we have learned from the observational studies and from the preclinical studies, that here in the middle, the interferon gamma could play a role in MAS. So if you look on the right side of my slide deck, you will see what does that mean in macrophage activation syndrome. The macrophage activation syndrome is when the macrophage is activated. Macrophage is a kind of a cell in our blood. When it will be activated, it will lead to what we know the cytokine storm. I think we have heard a lot about the cytokine storm now with COVID. And in this cytokine storm, we will have a cytokine hyperrelease. So we will have a lot of these cytokines as interleukin 1, interleukin 6, interleukin 18, TNF alpha, and so on. And each of them are responsible about the clinical presentation, some of the clinical presentation of in the macrophage activation syndrome. What we have observed from the preclinical and observational studies that interferon gamma is responsible about the activation, seems to be responsible about the activation of these macrophage. From the other side, what we have learned as well, that interferon gamma to be hyper-released, it requires a defective immune system. It requires an immune system that it is activated. So it requires a T cells and a natural killer cells, which are our immune cells, that are working in an abnormal way. They will hyper-release its interferon gamma that will trigger the activation of this macrophage. And we know to have this immune response activation, we need a trigger. And the trigger could be the underlying disease, as for example, the rheumatological disease. It could be the infection. It could be any genetical defect in the patient. So I hope, so with Sobi, we thought that one way potentially to reduce the activation of the macrophage is to stop the activity of this interferon gamma. To block the activity of this interferon gamma. And actually here in Sobi, we have a drug which is emapalumab. Emapalumab, it's a monoclonal antibody. So what does that mean? It means when you infuse this emapalumab in the blood, it will circulate in our system, and it will locate the interferon gamma. It will bind to the interferon gamma, and it will stop the activity of the interferon gamma, and it will stop also the activity of all what it is triggered by this interferon gamma. And Rashmi has spoken a lot, has mentioned a lot CXCL9, and we observe also from this preclinical and observational study that the CXCL9 is somehow also linked to this interferon gamma. So by blocking this interferon gamma, we will be able to reduce this CXCL9, and potentially we will be able somehow to control the MAS activation. This is the hypothesis we had here in Sobi. So that's why we started actually to investigate emapalumab in MAS, and especially in MAS when occurring in SJIA, in the context of SJIA. And the study, which has completed the enrollment today, is a small study. The role of this study was really to understand what's happening for these patients, if the drug is safe, if really interferon gamma and all what's resulting from this interferon gamma, CXCL9, CXCL9, can play a role in MAS, and if we really have a effect on the macrophage activation syndrome by blocking or by neutralizing the role of interferon gamma. And actually, the preliminary result support that yes, interferon gamma can have a role in MAS, and yes, by neutralizing interferon gamma, we can somehow manage and control this macrophage activation syndrome. But this is a small study, so nothing to be, as a conclusion cannot be confirmed unless we do a bigger study. That's why actually Sobi is going now toward initiating a bigger study where, again, we will be investigating the role of emapalumab in MAS when MAS is occurring in a context of stall disease. So on this slide here, you can see how this study is designed. In this study, we will have two phases. You will have the run-in phase and we have the interventional phase. The run-in phase, it's an optional, non-mandatory phase where we will be enrolling patients who are to be treated with glucocorticosteroid only. So these patients will not receive emapalumab. And we will observe what's happening with these patients under corticosteroid because there are still some medical questions, some scientific questions that we would like to understand better. And then if the patient is not responding to corticosteroid or not responding to a first-line treatment, he can then enter the interventional phase. And the patient in the interventional phase can come from the run-in phase as they can come from outside the run-in phase. So they can join the study directly into the interventional phase, any patient who is not responding to a first-line treatment. And this patient will have a treatment period with emapalumab for approximately 4 weeks, and then they will enter into a follow-up, and the follow-up will end 1 year after the last dose of emapalumab. And on a last note on this slide, I would like to say that this study today, it is on clinicaltrial.gov. I have here the NCT number, it's very small, but I hope you can see it. So please, if you are curious about the design of the study, feel free to go and consult clinicaltrial.gov. You will find all the details. The study has not started the recruitment yet, but we hope we can start in the months to come. With that, I end up my speech and I'm happy to take any question or clarify any misunderstanding or any concern. And thank you a lot for your attention.
Great. Thank you, Jeanette. And we do have the link that we're going to put at the end for both of the trials. So people will be able to get that. And then also it will be in the chat and we will send it. So to the replies. Now we did go a couple minutes over. So, but we do have some questions in the chat. So we're going to ask if you could check the chat and start to reply in there. Would that be all right?
Yes, now I need just to go to that. So I will stop sharing maybe because it's easier for me to go to that chat and then I will open the chat. If I manage to open it.
Okay. So while you're looking, you can type in some answers to everyone in the chat. We're going to move over to Sylvia. Hi, Sylvia. You need to unmute.
Hi, I'm trying.
There you are. How are you?
I'm good. How are you?
Great.
I'm trying.
Great. Wonderful.
Trying to share my screen.
Okay.
Can you see my slides or not yet?
Not yet.
This is always the part that gets tricky, right? Let's see. No. There we go. Should be, hopefully.
There we go. Yes. Yes.
Okay.
The winner.
Okay. Hello, everybody. I'm Sylvia Hanna. I am the Vice President and Head of Medical Affairs at Avalo Therapeutics. If you have done your research, you may have known us as Cerecor. So last week we changed our name and our NASDAQ ticker. So we are now Avalo Therapeutics and no longer Cerecor. But we are still investigating an IL-18 monoclonal antibody for Still's disease. So that has not changed. So we're a small biopharmaceutical company. We have 6 molecules that we are investigating in different rare disease, oncology, and immunology diseases. So we're small and nimble and mighty. We are driven by compassion and innovation in order to bring targeted therapies to patients in the immunology, oncology, and rare genetic disorders in those patients. So, and our development approach is really to look at predictive markers, and one of them is IL-18, as you've heard Rashmi and Jeanette speak about. And so right now we're doing clinical proof of concept. We're trying to prove that it actually works and then do larger trials. So that's where we are in the process. So today we're going to talk about Still's disease, as Rashmi has said, SJIA and Still's, same disease, different age groups. We'll talk about the correlation between IL-18 and Still's. We'll talk about our molecule that the drug doesn't have a name yet. It has letters and numbers. So it's ATVX, which is Avalo Therapeutics 007. So, all right. So as we know, adult onset Still's is a rare and not a very well understood disease. We're still learning a lot. It's in the United States, the estimates are 3,500 to 7,000, Western Europe, 3,400 to 6,900, and in Japan, 1,400 to 2,700. So there's no definitive genetic or infectious cause, and about 40% of patients suffer from severe chronic disease. And unfortunately, the treatment options aren't wonderful. There's NSAIDs, your typical over-the-counter non-steroidals, your Advil, your ibuprofen, Motrin, steroids, and then immunosuppressants, disease-modifying agents like methotrexate. And up and coming, we have monoclonal antibodies and proteins, anti-IL-1, anti-IL-6, and hopefully anti-IL-18. And as Rashmi, I really liked her slide with the garbage can. It is a diagnosis of exclusion. We haven't made much progress there. So basically, you have to have a negative ANA, rule out systemic lupus and rheumatoid arthritis. And when you've discarded all the other diseases, then you can be diagnosed with Still's, which is not a great way to diagnose a disease. There's an equal distribution between sexes. So some diseases, more women will be affected, more men will be affected, but here it's pretty equal. And then the age presentation is variable depending on whether, if you're pediatric, you'll get diagnosed with SJIA. So we talked a little bit about current treatments. And so initially, most patients are started with NSAIDs and steroid treatments, and there are some short-term and long-term side effects with these treatments. And then for patients that don't have any sort of improvement of their symptoms, they're called refractory patients. They are patients with a disease or condition that doesn't respond to treatment. Then you have the stronger drugs. So those disease-modifying agents like methotrexate or IL-1, IL-1 beta, IL-6, and anti-TNF antibodies, which we've heard people talk about today. And here are some of the names of the drugs that are currently used. So, and what's very, very important, as Jeanette said, you guys set me up really well, that patients have to be carefully monitored for the development of MAS, Macrophage Activation Syndrome. And while many patients do derive at least partial benefit from some of these treatments, some patients remain refractory, they don't respond at all, or they'll partially respond, and they're called partially refractory. So here at Avalo Therapeutics, we believe there is a need for more targeted therapies. And so we're targeting IL-18. It's, as Rashmi has said, and Jeanette has led up to it, IL-18 has really shown a critical role in various anti-inflammatory diseases. So right now we are targeting Still's. And so if you take a look at this, it's a little similar to the figure that Rashmi presented. IL-18 correlates with Still's disease activity. So if you look at this, in this study, 38 patients were diagnosed with adult onset Still's disease, 20 patients with SJIA, and then they wanted to look at patients, they took 65 adults and 23 children who had fever and or arthritis, but didn't meet the diagnosis for acute onset Stills or SJIA, and they served as their comparison group. And so if you look at the comparison group versus the patients that had the disease, you can see that IL-18 is almost 1,000-fold greater of elevated IL-18 serum levels. So we believe it's a significant and important biomarker in AOSD as well as SJIA. And so if we can find a biomarker, then hopefully we can try to decrease those IL-18 levels, and the disease hopefully your symptoms will decrease. That's the theory that we're using. So you can also find IL-18 in other inflammatory diseases because IL-18 is part of the inflammatory pathway. But again, unfortunately, AOSD has the highest. So compared to lupus or systemic sclerosis or rheumatoid arthritis or healthy patients, you can see that the IL-18 levels are markedly higher in AOSD patients. So based on that, and then also very, very similar again in systemic juvenile idiopathic arthritis, SJIA. And so here we have the diagnosis prevalence, again, diagnosis of exclusion, and IL-18 are significantly elevated. So if you look at SJIA active disease, and then patients with partial remission and patients with remission, you can see that when IL-18 goes down, so do the symptoms. And then these are our comparator groups. So monoclonal antibodies. Since we were in the middle of a pandemic still, we've heard a little bit about monoclonal antibodies. And what are they? Well, they're a type of medical treatment, and they're made in the lab, and they work like natural antibodies in your body to help you fight the illness. So it's been used in COVID, and we're looking to use it in Stills. So originally, AVTX-007, it will be the first-in-class anti-IL-18 monoclonal antibody. So it'll be the first. And it was originally developed by MedImmune and AstraZeneca, originally tested in patients with COPD. So chronic obstructive pulmonary disease, another inflammatory disease. And so I'm just showing this slide to show how initially when you're developing a drug, you have to figure out the appropriate dosage. So if the dose is too low, it may not be effective. If the dose is too high, you can have some safety concerns. So you have to figure out the appropriate dose in order to be able to give this to the patients. So here we were looking for an appropriate dose, and you can see that placebo is no drug, so that's up here. So obviously the IL-18 levels stayed high, whereas over here, 300 milligrams IV and 1,000 milligrams IV were the two groups. In those patients, you can see that the IL-18 dropped significantly. So, and IL-18 so far in our studies shows that it's pretty well tolerated. You don't really have serious side effects. Patients don't have side effects that lead to discontinuation of the study where they say, I can't take it anymore. I don't want to be on the drug. Obviously, patients can withdraw from study whenever they want. And then the ones that did have side effects, they were mild to moderate. One of them was worsening COPD, which is that's considered a side effect if your disease gets worse. The most frequent side effects, pretty common in most drugs, headache, worsening COPD, dizziness, very common, cough. If you have COPD, that's associated with cough, and then a slower than normal heart rate. Okay, so our study will be, again, if the study is successful, this will potentially be the first IL-18 monoclonal antibody. And so our study is in subjects with active onset Still's disease, and we're evaluating two doses, the two doses that I discussed, and it's in US and Europe. And so the primary endpoint is we're looking to reduce CRP, which is another biomarker. It's C-reactive protein. And we want to be able to see in lab data that this has been decreased by at least 50%. And we also want to see elimination of fever for greater than 48 hours. So that's how we would know that the study is successful. When the FDA evaluates studies, you have a primary endpoint, And if those patients reach this endpoint, then the FDA will say, your study was successful, we will approve the drug. And then there are usually some key secondary exploratory endpoints. And again, more lab data, change in IL levels, and obviously safety and tolerability. Is the drug safe? Even if a drug works, if you have safety concerns, then the drug will not get approved. So all of these things have to be met in order for the drug to be approved. And we are anticipating some initial data in fall of 2021. So we're very excited about this. And for more information, and I know that Tiffany has posted it on the website as well, you can speak to your physician if this clinical trial is appropriate for you and get more information at clinicaltrials.gov.
Great, thank you, Sylvia. That was fantastic. We have, I'm looking to see if there are any questions that are coming in. I do not see any of yet, but that does not mean they are not going to come in. I did want to thank you personally for explaining some of the words that are in there. The refractory is one. One of the things that we will do, in addition to collecting all of your questions, we will be working on providing answers to those, along with creating, we like to create glossaries so that we have that. That's one of them that's going to be on the glossary. So here we go. There's a question that can you see the chat, Sylvia?
Yes. So if in Medicaid remission and CRP is stabilized, would you no longer be eligible for the study? Again, there's multiple study criteria, and that would have to be evaluated by the study site.
Yes. Here, do you have any idea why some patients react so well to certain treatments and some don't? That's a pretty generalized question for everyone, I think. Did you have anything to weigh in on that specifically, Sylvia?
No, as a physician, that's a challenge with every disease, right? Some patients respond well, some patients don't, and we don't always know why.
And I think Rashmi touched on that a little bit too with the subgroup, and that's something that I could actually turn on my video. I'm sitting here speaking, and everyone's looking at my my stationary photo. That's something too that our organization, like the Systemic JIA Foundation, very interested in the whole subgroup. And we've been focusing strongly on working towards precision medicine and matching the right treatment to the right patient at the right time. And because with stills, it is a very unique, we actually say hashtag my stills because it is unique, and we do need to figure out what's best for each. And that's why we wanted to do this today as well, to show you what some of the research, some of the potential therapies for stills and complications of stills are actually in the process. So we are going to, you can still continue to ask questions inside the chat, and we are collecting them. If we cannot get to the questions, there is an opportunity for you to continue this conversation. And I'll be mentioning that here in a second. And I think Miss Katie is going to put a link in there for you, which I will mention how that works here in a second. So let me share my screen and believe I am sharing right now. If I'm not, I hope somebody will tell me.
We can see it.
Okay, thank you. So in sort of wrapping this up here in the conclusion, is the most important thing to remember when any therapeutic choice, but we're talking about clinical trials in particular, is the shared decision-making process. And always, first and foremost, education is the most important. You need to be informed, and that education can definitely come from your doctors, but it also can come from organizations. It can come from looking up research that's currently happening, and we hope all the organizations involved in this presentation today hope we can continue to connect you with those valuable resources. Always ask the why. Why is this important to me? Why could this potential, by joining a clinical trial, how could this benefit me? And we did mention earlier, a lot of times that you've heard it today, refractory. If your disease is not controlled with existing treatments. That's one of the top reasons why persons with our diseases tend to want to join trials. But you have to ask, what is the why? Why is this important to me? What could I benefit from? And weigh those pros and cons. Also, talk to your family members. So there's a lot of parents out there, obviously, who have to make these choices for their own children. And we encourage you to find other people who are in this situation and communicate. And again, I know Rashmi has a lot of resources for that, and wonderful conference as well, to connect with people. And then many of you are already on our Facebook Still's Disease group, the International Still's Disease Support Center. So you can find that on Facebook if you're not already. And there's a lot of people who you can speak with as well. And then, make your decision. You always want to include your treating physicians and anyone else who's important in your therapeutic decision making. This is where the link in the chat is going to come up and become handy here. So Miss Katie has put a link, and we can keep the conversation going. So our organization has a platform called AiArthritis Voices. If you are a person living with Still's disease, SJIA, or adult onset parents of juvenile patients, caregivers of adult patients, you can sign up to join this. Well, all stakeholders can sign up to join, but for this particular continued conversation, you sign up to join, and we have an online community set up. For the first time, we're launching it with the Still's Disease Project, and you can actually continue the conversation. We'll have representatives from the Systemic JIA Foundation in there, Autoinflammatory Alliance, also Sobi and Avalo, all will be in there, will be in there. And so you can continue the conversation with people who have been in here. You can continue to ask questions. And on September 7th, which is Still's disease, we're going to have a rewatch party. So we're going to rewatch portions of this, and we're going to invite persons living affected by Still's disease to join. So it'll only be the patient voices only. And we're going to review snippets of important parts of this webinar, and then you'll have an opportunity to stop and communicate and ask questions and just really dive in. We're here to give you the information that you need, and that, I think, is what the biggest takeaway is today. We have another about 8 minutes or so left, so I am going to turn this over back to the panelists. If anyone else wants to put your cameras on, we'll put the panelists back on. And if anyone has questions, we can put those in the chat. And I'm going to actually also ask Miss Katie if any questions have come in while I was talking, because I wasn't looking.
Sure. Well, there was one that was recent. I've recently been asked by my rheumatologist if I'd be open to taking 2 biologics. Is that something that people with AOSD and other autoimmune conditions can do?
To be on multiple biologics?
Yes.
We do know of several patients in the SJIA group, as somebody else also posted in the chat, who are on multiple biologics. It is really done one at a time, one case at a time, and in consultation with the specific rheumatologist, but it is not unknown. Another place where you'll find experience with this type of combining biologics is actually in the autoinflammatory group, where there are several patients with genetic autoinflammatory diseases who are on long-term Kineret and on long-term Ilaris, and then are taking Kineret for flares. So those are the 2 places you might want to get more information about it.
Thank you, Rashmi.
Actually, one more thought about that. There's a case study if you search on PubMed, there is one case study of a patient with SJIA who was put on both an IL-6 and an IL-1 blocker. That might be something you can take to your rheumatologist.
Great, thank you. And then as far as also the question of recommending talking to your primary care physician, given such a rare disease, one of the things that we're going to send out is actually some tips on opening the communication from our organization. And then also, I know, Rashmi, you all have wonderful resources on your website. as does the Autoinflammatory Alliance. And our organization has patient-reported Still's disease brochures on our site. So we can definitely follow up and make sure that we provide everyone with links. Those are wonderful to take to primary care physicians to learn more. I think we have more questions that came in. I'm trying to look up here. Okay. I think those pretty much wrap up our questions. I do want to put the other screen on because this has all of the links. We have the links to the clinical trials that were talked about today. We have the direct link to join the AiArthritis Voices platform. And then also just wanted to thank really everybody who has participated. We have had an amazing interaction here. All of the panelists, thank you so very much. We will be, again, downloading all of the questions that were asked during this chat, and we will follow up with everybody who has attended and wasn't able to attend with a link to watch this. And you can participate in the continued conversation if you'd like in this new platform. And we will send a sheet of questions with as many answers as we can back when we do know those. So thank you, Rashmi. Thank you, Jeanette. Thank you, Sylvia. I think we are signing off for today, and we want to thank everybody so much for participating.
Thanks.
Thank you.
Thank you.